Hi everyone, this is Amrita and welcome back to Biology 101. Let's start with the topic which we left in the last video. So, laws of inheritance. There are three laws given by Mendel: law of dominance, law of segregation, law of independent assortment. Now, I'll break down all the three laws for you. The law of dominance and law of segregation we'll learn now because it is based upon monohybrid cross. That is, the only one character is used in the hybrid, which we learned in the last class. And independent assortment will depend on dihybrid. That is two characters. So that will come later. Today, law of dominance and law of segregation. And then we have incomplete dominance also. That is a special case. Okay. So what is law of dominance? Here characters are controlled by units, that is factors. I already said in the last class, factors now known as genes. Previously factors was the word used by Mendel and now it is known as genes. So these factors occur in pairs. In a pair of factors we know there is dominance and recessive, that is the alleles can be dominant or recessive. It's used to explain the expression of only one character, that is only which is dominant will be shown on the phenotype in F1 and both F2. In F2 we can see both, but in F2 major is, that is 3:1 ratio, major will be the dominant one. Okay, so law of segregation based on the fact that alleles do not show any blending and both characters are recovered. That is law of segregation. There is no blending as said in the previous class. There is no blending, meaning there is no nothing in the between. There is only either dominant or recessive and both the characters are recovered like that. Parents have both alleles during gamete formation, but alleles segregate so that a gamete can receive only one of the two. That is, we have either we'll have T and T, both will be there in the parents, but in the gamete only one will go, either big T or small t, so that there is no blending. Now homoparent, if the parent can have two alleles, right? Now if the parent is having one type of gamete, that is T, big T, that is, or small t, both come under homoparent. If it is big T and small t, they come under heteroparent, that is two types but in equal proportion. Okay, so next is incomplete dominance. This is a special case under law of dominance. Now in some experiments, sometimes F1 had progenies which do not resemble either one of the parents. That is, see, now we have either, we already saw monohybrid cross, right? There in the F2 generation, either we found tall or dwarf, that is 3:1 ratio. 3 will be tall and 1 will be dwarf. But now in some experiments, scientists found that in the F1 itself there is no tall or no dwarf, that is something in between. So the example we have dog flower, snapdragon, antirrhinium. Don't worry, all these three are the same plant. You need just you need to remember the all the three names. Now here we have textbooks diagram, that is they have taken this is the dominant one, capital, right, dominant. This is the recessive one, red is dominant, white is recessive. They are taking the gametes, they are fusing it. Now F1 generation we have pink, not red, not white, but pink, that is, this is blending, which means there's no segregation here. So this is the rare case. Now what, now if we find a pink flower, we don't know the parents, right? So what we do, we self the RR, the small, big R and small r. When we self it, we will find one red, one white and two pink, that is 1:2:1. This is the ratio for phenotype as well as genotype. So this ratio is important because now they might ask in question, which case gives both phenotypic and genotypic ratio same. We should give the answer as incomplete dominance. That is the example for dog flower. Then explanation of the concept of dominance. This is also from textbook. What is exactly dominance? Every gene contains info to express a trait. In diploid organism, two copies of each gene is there. It can be either identical or non-identical. Identical means homogeneous, like capital T or small t. Non-identical will be capital T and small t. Now example, gene that contains info for producing an enzyme. Now like we'll get curiosity, right? How is the dominant truly, how is the gene or allele truly dominant and other one is recessive? How does it become recessive? Now let us take an example. This is also from the textbook only. Gene that contains info for producing an enzyme. Okay, that is the topic, like that's the example which we are taking. Now it has two allelic forms, that are the copies. Normal allele will be there or the modified allele. Now normal allele produced normal enzyme, that means that is dominant, which is normal is dominant. When it is modified, there can be three forms, that it can produce normal or less efficient enzyme, it can be non-functional enzyme, it can be no enzyme at all. So when there's normal or less efficient, let's take its normal only in the modified itself, which will be equivalent to the unmodified allele, which means it can produce the same trait. So if we say it can be TT itself, the normal allele will be T, this also will be T when it produces the same trait. So it will be homogeneous TT. Now if this produces normal will produces capital T, and modified non-functional enzyme or no enzyme at all, it will be small t. So capital T, small t, small t will become recessive and capital T will become dominant. That is why here non-functional enzyme or no enzyme phenotype may be affected. That is why generally recessive. Now recessive trait is formed due to non-functional enzyme or no enzyme is produced. That time only recessive is produced. Otherwise we have dominant only. So this is all for now. Now the next topics we will have codominance. After that inheritance of two genes, dihybrid and all will come. So we will see that in the next